university's programs for the academic year, and every academic who rises to the rank of professor in his or her career in the University of Ghana is expected to deliver an inaugural lecture. This offers the university the opportunity to recognize and showcase the academic achievements of faculty, and also enables professors to share their research with colleagues both within and outside the university, while celebrating an important personal milestone with family and friends. It's now my distinct pleasure to introduce the chairperson for today's inaugural lecture. Our chairperson is the first female vice chancellor of the University of Ghana. She is a professor of linguistics and an experienced university administrator. She has over 20 years experience in the higher education sector, and she's built her experience in governance through attendance of management and leadership programs, and also by on-the-job training. She's skilled at change management, and she brings a lot of innovation and resourcefulness to her position as Vice Chancellor. She's a member of many prestigious societies, too many to mention here, and she's a fellow of the Ghana Academy of Arts and Sciences, and also the founding president of the African Humanities Association. Distinguished lecturer, ladies and gentlemen, please join me to welcome Professor Nana Appiah Amfu, Vice Chancellor of the University of Ghana and chairperson for today's lecture. Thank you.
Thank you, Madam Registrar. Pro-Vice Chancellors, Registrar, Provosts, Deans, Directors, Heads of Department, our distinguished lecturer, Professor Solomon Fiifi Ofori Acquah, family and friends of the lecturer, faculty staff, and members of convocation, past and present senior officers of the university, alumni, students, and friends of the university, corporate executives, members of the diplomatic corps, if there are any, esteemed traditional rulers present, Sangana, the paramount chief of the Sangarugu traditional area, is present with us, and the representative of the Osu Manche Ni Note Owo IV. Our friends from the media, invited guests, distinguished ladies and gentlemen, those who are present here and those joining us virtually, good evening to you all. Once again, I have the singular honor and pleasure of chairing another inaugural lecture, the third in the series of inaugural lectures scheduled for this academic year. I am particularly enthused about this evening's lecture as it highlights the research work and contribution of the first professor of medical laboratory sciences at the University of Ghana. Today's lecture will focus on a very sensitive health issue, sickle cell disease, or SCD. In the early part of my relatively short life, I used to hear people use derogatory terms such as sickler, and I'm sure that many of you can relate to that, when referring to people who are suffering from this inherited genetic disorder and have often had to endure the unrelenting pain arising from a crisis situation. Thankfully, due to the prevalence of sensitization and education, this narrative is gradually changing, though we are not there yet. Today, we are privileged to have Professor Solomon Fiifi Ofori Acquah, one of few scientists with specialization in this subject area, whose academic journey has given him the unique opportunity of working in both the Western world and Africa to share interesting but revealing perspectives on the evolution of scientific inquiry into sickle cell disease during his inaugural lecture. Throughout his career on a journey to find a cure for sickle cell disease, Prof. Ofori Acquah has made a number of invaluable contributions through research and advocacy to promote knowledge about the disease. These interventions include creating the Sickle Cell Disease Genomics of Africa, Sickle Gen Africa Network, a multinational project serving as a catalyst for transformative change in genetics and sickle cell disease in Africa, equipping health professionals in Ghana and the sub-region in genetics with requisite knowledge in sickle cell disease, establishment of the West African Genetic Medicine Center, WAGMIC, an African Center of Excellence for capacity building in genetic health, and the development of a potential cure to sickle cell disease through in vivo gene therapy. Distinguished ladies and gentlemen, what a relief it would be to finally find a cure to this disease which inflicts so much pain on its patients. As a research-intensive university, it would be even more gratifying to have one of our very own at the forefront of this groundbreaking intervention, which will benefit the entire human race. Now, at this point, kindly permit me to share a brief profile of our distinguished lecturer. I say it is a brief profile, yet you would have to be patient with me. But the full version of the profile is in their brochures, and we have both hard copies, and you can also access these online using the QR codes provided. So our lecturer for this evening, Professor Solomon Fiifi Ofori Acquah, is the first professor of medical laboratory sciences at the University of Ghana. He is a former dean of the School of Biomedical and Allied Health Sciences, in fact, the immediate past dean, and the founding director of the West African Genetic Medicine Center. Professor Ofori Acquah was born on Friday, 15th March, 1963 in Cape Coast. He has two older children, Araba Ofori Acquah and Jake Ofori Acquah, and he's married to Miriam Sidiki, and they live with their son, Solomon Ofori Acquah Jr. Professor Ofori Acquah had his ordinary level secondary education at Adisadel College, Cape Coast, as well as his advanced level general education at the same institution. He attended Bromley College of Technology, Kent, England, graduating with a higher national certificate in medical laboratory sciences in 1989 and part 1 of the fellowship of the UK's Institute of Biomedical Science in 1990. He enrolled in Birkbeck College, University of London, to read biomolecular organization and graduated with a Master of Science degree in 1992. He completed formal education at King's College School of Medicine and Dentistry, University of London in 2000 with a PhD in molecular genetics. He obtained a certificate in leadership in 2012 at Emory University, Atlanta, Georgia, USA. Professor Ofori Acquah's career started as a trainee medical laboratory scientific officer in hematology and blood transfusion at Farnborough General Hospital, Kent, England. He became certified to practice clinical laboratory hematology and blood transfusion by the UK's Council for Professions Supplementary to Medicine in 1989. He switched to a research career at the Department of Hematological Medicine, King's College Hospital, London in 1994, and parlayed his project a year later into a PhD thesis. After completing, he was appointed Scholar of the Comprehensive Sickle Cell Center in 2001 and promoted to Assistant Professor of Cell Biology and Neuroscience in 2002 at the University of South Alabama. He joined the University of Ghana briefly as Associate Professor in the Department of Medical Laboratory Sciences from 2011 to 2012. He moved to the University of Pittsburgh, also in the USA, in 2013 as Associate Professor of Medicine and Human Genetics, and currently holds that position as a part-time faculty. He assumed office as Dean of the School of Biomedical and Allied Health Sciences at the University of Ghana in March 2017, and his term ended last July. In August 2022, he was appointed Director of the West African Genetic Medicine Center, which was established initially as an African Center of Excellence in January 2019, with him as the center leader. Prof. Ofori Acquah has developed many educational and training programs and taught and mentored students across the academic spectrum in multiple institutions in Ghana, Europe, and the USA. He served as mentor for junior faculty in the Summer Institute program to increase diversity and the program to increase diversity for individuals engaged in health-related research in the USA. He designed the Pittsburgh Undergraduate Research Diversity Program and the Pittsburgh Intensive Training in Hematology to promote hematology research among junior investigators. He secured funding under the R25 training grant mechanism of the National Institute of Health, USA, to start training programs in the University of Pittsburgh. As a visiting lecturer at WACBEP, our own WACBEP, he designed and taught a graduate genetics course of the center. More recently, he designed and is the coordinator of the MSc Genetic Counseling Program offered by WACBEP. Professor Ofori Acquah was recipient of the Outstanding Postdoc Mentor Award at Emory University in 2012. Professor Ofori Acquah has conducted research principally in 4 areas, namely genomics, vascular biology, breast cancer, and sickle cell disease, over a career spanning nearly 30 years. His research in the late 90s in genomics defined DNA sequence variation that influenced levels of fetal hemoglobin among sickle cell disease patients. As a postdoctoral scientist in the early 2000s, he helped to establish how multiple transcription factors and drugs control fetal hemoglobin production in sickle cell disease. He began independent research in his own laboratory in the mid-2000s with a $1 million grant from the NIH. He was focused on the role and mechanism of activated leukocyte cell adhesion molecule in tethering endothelial cells together in blood vessels, and so on and so forth. Professor Ofori Acquah's work in endothelial cell biology and vascular permeability catalyzed his most impactful research to date. He's published in the he's published his research in many journals, including the prestigious Journal of Clinical Investigations, and has received over 230 citations to date on this particular research findings. Professor Ofori Acquah has expanded his research on extracellular heme to develop 5 separate multi-million-dollar projects to date, and they are all funded by the NIH. Professor Ofori Acquah assembled a multidisciplinary team of more than 30 researchers in Ghana, Nigeria, Tanzania, South Africa, and USA to work on the Sickle Gen Africa project. This team has already chalked up important milestones. Professor Ofori Acquah is leveraging his whole genome sequencing project to build capacity in all aspects of genomics at WACMIC in collaboration with a major US academic partner. In addition, he has recently designed a new project in partnership with a multinational pharmaceutical company on gene-based cures for sickle cell disease. Other areas of current research include several of his MSc Genetic Counseling student mentees, and they are focused on defining societal, policy, and infrastructural barriers to integration of genomic medicine into clinical practice in Ghana. Professor Ofori Acquah has authored over 80 research papers, reviews, and an extensive leadership program at Emory University. He obtained experience in leadership as director of the Center for Endothelial Cell Biology, Emory University, director of Allied Health Sciences, and most recently as director of WACMIC, both here at the University of Ghana. Since May 2022, he has been the president and chief executive officer of the Sickle Cell Foundation of Ghana. He was vice chair of the American Society of Hematology's Committee for Promoting Diversity. He is on the editorial board of Blood Advances, a journal of the American Society of Hematology. He served as guest editor for Frontiers in Genetics and has reviewed transcripts or manuscripts for over 30 scientific journals. He's a founding executive member of the Ghana Biomedical Convention, and he was vice president and president of the organization. He received an appreciation award in 2016 for his role in founding the Ghana Biomedical Convention. Professor Ofori Acquah has presented in over 100 conferences, workshops, and scientific retreats across the world. I told you have to be patient with me with this brief profile. I'm almost at the end of it. Professor Ofori Acquah has served on multiple national and international committees and boards. He's consulted for the Foundation for Research Development, Federal Research Development at the Federal University of Minas Gerais and other universities. Professor Ofori Acquah is a member of the Board of Directors of the University of Ghana Medical Center and the Sickle Cell Foundation of Ghana. Ladies and gentlemen, please relax and enjoy the lecture as Professor Solomon Fiifi Ofori Acquah takes us on a journey with sickle cell disease around the world and a return home to help find a cure. Thank you.
Go. Asida ne ade manyankopon na dumu na nanu matra nkonwa mi jira shampoo ya na mu akuni ni anintre mama munyina akwaba yenyina nim adekodi enti ya hyiamwa asuapon ya nyamfia duosonum abribia adura aya abadwam kasa kire ya yiwe wo bia wonimede enipa bia mansani no agogovie fufua akromasha no nson na no mu ne awewe ya ba mra no nyamfo bi ne yehunu no na ne din aye Professor Solomon Fiifi Ofori Acquah ogwa kepkos no frɛ wodi wa na obusa sa fikesie mu kwan wan ebe kyerɛ ɛyɛ aduanani aduana twea brade apɔtra wɔ me nsuo na ne na nanom aduanafoɔ deɛ aduane nyɛ wɔn na twenu amponyame ɛyɛ aduanani aduana bae kofi abrenu nie moni adwuma pa wayɛ adwuma somomen Ghana mebi adua bibi manso yate wo nkawa ma no ne ho asɛm deɛ ame sɛ Prof bɛgyina no waka ne nyinaa ɛdɔn ne bladru sɛ wɔmmra na wɔmmɛkasa aduana ba kofi amansani no abɛno aduro bɛkasa yasoma mi o sɛ memfa sepre wa mɛgye wo taataa yasoma mi o sɛ memmra wo na mema frɛfrɛ wo sɛ nyankontom frɛ nsua no ba tututu yɛ ma me Esther Kwansa ɛna ne ba no owura akyɛ Ofori Acquah awo ne bɛɛ aduana ba wo brɛ nie kofi yɛ dɛn aso sɔre yɛ ma mo so Bama bae ablanu adro aja kasa o me di sepre we frefra ma musu e aduana ba wo blanu aso Ghana tia wo
Madam Chairman, the Vice Chancellor of the University of Ghana, Pro-Vice Chancellors of Academic and Student Affairs and of Research, Registrar, Provost, Deans, Directors, and Heads of Department, faculty and staff and members of Convocation, senior members of the University of Ghana, past and present, alumni, students, and friends of the university, corporate executives, government officials, our esteemed traditional leaders present, Sagnana, it is really an honor to see you on the front seat. Thank you so much for making this already an honorable occasion. Eminent clergy, friends from the media, invited guests, from the Palace of the Osu Manche, thank you for joining us. We really appreciate this. Distinguished ladies and gentlemen, it is really a singular honor to be here to give an inaugural lecture. The Vice Chancellor, who's chair, doesn't know this, but I had opportunity to do this in a university in the United States, and I kept postponing it because I wanted to do it here first. What I'm going to take you through is a scientific journey that I began several years ago. It is a journey trying to understand sickle cell disease, just so that it happened in the context of traveling around the world. So here's the outline of my presentation. Given that sickle cell disease is central to what I'm going to talk about, I want to give you a brief preview of the state of sickle cell disease in Ghana. Then I've broken my talk into three parts: the scientific journey with sickle cell disease around the world, and a return home to help find a cure. In part one, we will talk about our efforts in pursuit of genetic factors and pathways that can reduce the severity of sickle cell disease. In part two, I share with you the fun we had in designing animal models to better understand the disease. And then part 3, which is probably the most important for me, is the return home to build capacity in translational genomics with the aim of facilitating gene-based therapeutics right here at home. What I'm showing is the map of Ghana. And on this map of Ghana are names of sickle cell disease by various people in this country. The Gas call it Chwe, the Ashantis call Hututuo, the Wala call it Gurugurihi, and the Gombes call it Daminuhi Dono. These are the names Ghanaians have always used to describe sickle cell disease, long before we even knew there was a thing called a gene. So you see, when it comes to the clinical presentation of sickle cell disease, our ancestors have known about the disease for a very long time. We've known about this disease for a very long time. In talking about the state of sickle cell disease in Ghana, it will be remiss of me not to highlight the Herculean work that Professor Ohim Frimpong has done. A lot of what we know about sickle cell disease was due to this one man, Professor Kweku Ohim Frimpong, who had a vision to create a situation where everyone with sickle cell disease or its related conditions in Ghana is properly diagnosed and offered education, counseling, and modern medical psychosocial services. Ohim Frimpong finally is coming home to rest this week, and his may soul rest in perfect peace. Through his efforts, the map you're looking at are regions in Ghana where we have screened newborn babies for sickle cell disease. Virtually everywhere in this country has been touched by capacity building to screen newborns for sickle cell disease. We haven't quite gotten there yet. About 900,000 births in Ghana every year. And I will show you data that of those births, 1.7 will have sickle cell disease. And yet we don't have a national program to screen for sickle cell disease. From 1995 to just last December, the Sickle Cell Foundation of Ghana, through the work Ohim Frimpong did, screened over 500,000 babies. 500,000 babies have been screened within that time. And from that, we know that the incidence of sickle cell is 1.7% in Ghana. This work was not done entirely by the Sickle Cell Foundation of Ghana. This work was done in partnership with the Ministry of Health, the Ghana Health Service, our own Noguchi Memorial Institute, where the laboratory work was done, and countless of nurses and midwives and pharmacists who are taking the fight to sickle cell disease. So I don't want anyone to think the Sickle Cell Foundation of Ghana claims all the credit for this work. No, it's been done in partnership with these folks. Ohim Frimpong also managed to convince the government to work with Novartis to develop a public-private partnership to test an idea. You see, those of us who've had the opportunity to work abroad know what good quality services look like. And oftentimes when we return home, we simply want our fellow Ghanaians to have the same level of services. Hydroxyurea is a drug that works for sickle cell disease. And through this PPP, hydroxyurea was introduced to Ghana with support from the government. I have to say, we have kept on relentlessly to convince government to make good on its promise to make hydroxyurea available. Now, don't ask me what I was asking the vice president in that conversation, but I was probably asking him, you promised that you get hydroxyurea to sickle cell patients, so please, sir, can you honor your promise? A month after that picture was taken, the chief executive of the National Health Insurance Scheme announced hydroxyurea freely available for sickle cell patients in Ghana. This is the only country in Africa where this is happening. So we've established centers all across Ghana for physicians to become competent in using hydroxyurea, because hydroxyurea is a myelotoxic drug. You can't just put a patient on it and just leave them alone. You have to monitor them. We did this through an implementation project with Novartis to demonstrate that here in Ghana, sickle cell patients can take hydroxyurea safely. The physicians have the competencies to monitor them safely. And after years of doing that, this was what pushed the government since July 1st last year to make hydroxyurea available. The chart you're looking at is the number of patients who are on the hydroxyurea therapy. We monitor these patients. And even though the project ended in 2022, we are continuing to monitor these patients. And you can see the first quarter of 2023, 4,557 patients are on this program. There are more patients taking hydroxyurea who are not captured in this graph. So, Madam Chair, even though the burden of sickle cell disease and the patient experience remains a major developmental challenge in Ghana, newborn screening, though, is becoming more widespread. And this is being coordinated by the Sickle Cell Foundation of Ghana with government and non-government agencies. As I just showed you, hydroxyurea, which is the #1 drug sickle cell patients take anywhere in the world, is now on the essential drug list of the Ministry of Health and is freely available. If you're a sickle cell patient and you go to a registered physician, they'll be able to prescribe hydroxyurea for you. So, Madam Chair, the state of sickle cell disease in Ghana has improved remarkably over the past decade. In fact, the Ministry of Health even has a very nice booklet that helps and trains physicians in how to properly manage sickle cell. So we're doing a very good job of helping these patients. But what that means then is that in the next 10 to 20 years, the number of sickle cell surviving patients is going to go up. We are doing newborn screening, we're giving hydroxyurea, so inevitably, more sickle cell patients will survive. It is our duty, therefore, to look for better ways to manage sickle cell patients and to join the effort to ultimately find a cure for this disease. It's our moral duty to do that. Years ago, when I was a PhD student, it was this same level of vigor and enthusiasm that pushed me to go into the lab even when it was snowing back in London. I was in pursuit of genetic and other pathways that can help explain the severity of sickle cell disease. This journey started in London as a PhD student. I followed the same quest in Mobile, Alabama, and in Pittsburgh. And the thing about science is you do work in your lab, and then you have to get out of your lab and go and talk to other people. So the arrows you're seeing is me jetting around, discussing the work either in conferences or in workshops. And sometimes when you tell somebody else the work you're doing, they may even ask you a question that will help you to develop this further. I have to apologize. The next section is going to have some scientific terminologies. You're going to hear me say polymorphisms. There is no chi word for that. So you have to bear with me. We will talk about whole genome microarray. And we'll talk about humanized mice. Mice that have been humanized. Don't get scared. Was look fetal hemoglobin we should make. Very simple. But in molecular biology, we say it's localization of 6 regulatory elements at the beta globin locus. We're just looking for a small piece of DNA and seeing whether the sequence there is important. This is a schematic of genes. Don't get worried about it, it's just genes. The genes that code for proteins that we are aware of. Our interest was trying to find out whether sequences, DNA sequences, that are very close to the gene that makes fetal hemoglobin, and sequences that were far away, but we know had some influence. Which of those 2 sequences was the most critical? That is all this was about. DNA sequence variation in the beta globin locus that is linked to different types of sickle cell haplotypes. The experiment was relatively easy. And one of the things I'm hoping will come through this lecture is how some of the experiments in the early stages of my career were fairly simple and relatively mundane to the kind of work that we're doing right now. Here, through the efforts of sickle cell patients who volunteered for our study, we took blood from them. As we substantiated that finding with another study that showed essentially the same thing. So I'm not going to go through this one. Keep in mind that the whole purpose of these studies were trying to find out things in our genome or our DNA that can make one sickle cell patient not be very sick and another one very sick. In my PhD thesis, I studied siblings in London with sickle cell, both with sickle cell, same father, same mother. One is in hospital almost every week, and the other one is playing football and doing other things and not being sick. So there has always been an interest to find out what else controls the clinical expression of sickle cell disease. And this factor, NRF2, had become important, and so we did studies on that. Our goal here was to determine whether this particular pathway was involved in the progressive severity of sickle cell disease. We know that all babies are born with completely new, brand new organs, new heart, new lungs, everything is new. And when that baby is born and has sickle cell disease, and another baby does not have sickle cell disease, their anatomy, the lung, the heart, everything looks exactly the same. But as the sickle cell disease begins to manifest, those organs get to become sick. And so what I was interested in here was, what are the pathways that are involved in that age-related deterioration in the clinical expression of sickle cell disease? And NRF2 became important. Now, I just didn't pick up NRF2 just because it has a wonderful name. We had done work earlier where we looked in the entire genome. Okay, I promise you there will be some technical terms here. So here we looked at the entire genome. Global gene expression profiling. We took cells called endothelial cells. These are cells you are very familiar with because you can all identify your blood vessels, right? You all know where your blood vessels are. Your blood vessels are basically tubes of cylinder. In the internal diameter of your blood vessels are these cells we call endothelial cells. They line the blood vessels very nicely. So we took these cells and we exposed those cells in culture to heme. You hear a lot about heme. It's a byproduct. It's something that is released when red cells get damaged. And in sickle cell disease, a lot of red cells get damaged. And so we took this heme, exposed it to these endothelial cells, which you can now think of as a blood vessel on a petri dish in a lab, and ask, how do these cells react? But we were not looking at one pathway. We were looking at all the pathways in a cell. And we discovered that they react by either increasing or decreasing expression of genes. The fancy word we use, upregulation and downregulation. Basically, up or down. So you're looking at something we call a heat map. The green is down, the red is up. And the gene at the very top is a gene called hemoxygenase 1. It became very clear to us in this simple experiment that this NRF2 was at the center of almost all the responses that were important. So this is the reason why we wanted to find out what NRF2 is doing in this project I'm going to discuss with you. And this study that we did was very interesting. We studied transgenic sickle cell mice. These are mice, experimental mice, where the gene that codes for hemoglobin has been swapped and the human gene put in that mouse. So that mouse makes human hemoglobin. And when it's making human hemoglobin S, it has sickle cell disease. It gets pain, although the mouse can't really tell you it's in pain, but you can figure out that the behavior of the mouse. Remember I said that all our organs are brand new at birth, same for the mouse. With time, the mouse's organ deteriorate, just like patients. So we decided to do a longitudinal study. We enrolled 80 mice, and trust me, they volunteered for this. We enrolled 80 mice and just followed them for 10 months. A group that has sickle cell and a group that did not have sickle cell. And we didn't bother them too much. We only went to them every month to ask permission to take a blood sample. That's all we did. For 10 months, nothing else. Every month we go, we take blood, wait, we take blood. And then we're also monitoring how they are doing. So we're not doing anything to them. The first graph you're seeing is called a Kaplan-Meier curve. Again, nothing fancy. It's just a survival curve. And you can see that at the very top, mouse that we are calling AA. These are mice that are making normal human hemoglobin. And you can see the 10 months we followed them, not a single one died. Whereas the SS, we lost 4 mice within that 10-month period. We then looked at the blood that we took to see what is their anemia like. Total hemoglobin simply means anemia. And you can see that the normal mice, the AA mice, their hemoglobin for 10 months basically stayed the same. Whereas the sickle cell mice, there's a 25% drop when they got to about 6 months, and that stayed down. This gave us an insight into what happens with aging. Keep in mind, we haven't done anything to these mice. All we've done is taken a sample every month. And so we looked in the blood and we see that when red cells get crushed and they release hemoglobin, it stays in the plasma. And so you can see that whereas there is no increase in the plasma concentration of hemoglobin in the normal mice, you see it going up in the sickle cell mice. TPH is another marker of hemolysis. And the last one, LDH, is another marker of hemolysis. What this slide is telling you is that we found that with aging, without doing anything to these sickle cell mice, they hemolyzed. And their hemoglobin kept dropping. The question was, all these events in the vasculature, the hematology, was the hematology going to have any effect beyond the blood vessels? We had a particular interest in the lung, so not surprising, we looked in the lung. And if you follow this, these are the lungs that we have taken out. We have stained them so it's easy to see. As you know, lungs is where the air goes for us to get blood oxygenated. So there are lots of air sacs in the lung, which is why you're seeing this. But if you keep your eye, you see that by 10 months, the sickle cell mouse lung looks very, very different. It has chronic lung disease at 10 months. And because we're interested in this gene called NRF2, we're able to do a little trick. There are mice that you can make. The reason why we use mice for experiments is that if you're interested in a particular thing, you can remove it in the mouse. We call it knockout. Okay? So you can knock out any gene. So for us, it's not going to kill the mouse, right? So we took a mouse that had the NRF2 knocked out, and we transplanted that mouse with bone marrow from a sickle cell mouse, making that mouse now a sickle cell mouse, but with the NRF2 knocked out. I only want to show you one image. So the wild type SS mouse is the normal SS mouse, and the NRF2 normal mouse is the SS mouse that we have created in the background of having no NRF2 activity. Now, you don't have to have a PhD in molecular genetics to appreciate that the one to the left looks pretty clean and the one to the right is not. It is congestion in the pulmonary vasculature with red cells. We concluded, therefore, that NRF2 was important in keeping sickle cell patients' organs not getting sick. This then meant that NRF2 is a potential target that can be used to develop therapeutics to keep sickle cell patients nice and safe. At the same time, we're doing collaborative studies with a colleague of mine, Markel Dubon, who was at Vanderbilt, And following the same paradigm, we wanted to ask, one of those NRF2 targets is an enzyme called hemoxygenase 1. Its job is to break down heme. We wanted to ask whether the amount of hemoxygenase 1 you have matters. But the way we are asking it is genetics. So we are looking at the gene for hemoxygenase 1. There are some sequences, nothing fancy, that are different in you and me. As we sit here, 99.9% of our DNA sequence is identical. But there is 0.1% that is different. It is that difference that can make somebody get sickle cell disease. It is that difference that can make somebody take a drug that has worked very well for his cousin who had prostate cancer, and he has taken the same drug and it's not working, because we metabolize drugs differently. So here we're asking, the small differences in the promoter of this gene, does it make a difference with respect to the risk of sickle cell patients getting acute chest syndrome? And the short answer is that yes. The way this repeat works, as you all know, we inherit one group of genes from our mom, one group of genes from our dad. And so in this experiment, we are defining sickle cell patients by whether or not they have inherited a small allele from mom, a small allele from dad, which is why we call it SS. SS here is not sickle cell though. SS here is short. So you can see that the sickle cell patients with two short alleles, their rate of hospitalization for acute chest syndrome was very, very low compared to all the other variations that you can get. This made a lot of sense. And so then what it meant was that sickle cell children who carry two short alleles are protected from acute chest syndrome. This fitted again with the studies we did with NRF2, showing that in fact this is a potential target for developing drugs. And a fellow in my lab did similar experiments and confirmed, this time in another group of sickle cell patients. One of the benefits of the traveling is you meet people and you talk to them, you present. This work I'm showing you came out of a conversation in Strasbourg. I had just given a talk. There was a French guy who came to see me. He was very excited that I had all these mice. And he had questions he wanted to ask, and he didn't have the mice. And I casually said, oh, we have lots of mice. You know, if your student wants to come to Pittsburgh, it's not a problem. And then I left. And 3 months later, I got an email. Emmanuel is coming next week. So Emmanuel Sharon joined my lab as a visiting PhD. And they were interested in exercise. Don't adjust your vision. That is a sickle cell mouse on a treadmill. We studied these mice with Emmanuel's PhD to ask a very simple question. And the answer was that moderate exercise reduced inflammation. It reduced the number of white cells. It actually increased the proportion of hemoglobin that is oxygenated. And so through this experiment, we were able to conclude that, in fact, moderate exercise is good for sickle cell patients and potentially can also reduce their disease. So, Madam Chair, I want to summarize part one. That in our pursuit, looking for genetic factors that can lessen the severity of sickle cell disease, we're able to define which region on the beta globin locus is more important. We identified a pathway, NRF2, as a major modifier of sickle cell disease in terms of the progressive deterioration of the disease. And we also show that, in fact, moderate exercise is a good thing for sickle cell disease. Now, in part two, I want to focus on our efforts to try and develop better models to understand the disease. Most of this work was done in Atlanta, Georgia, and later in Pittsburgh. And again, the same idea of going around the world, discussing the work, and sometimes getting better ideas. The terminologies here are not that many. You've already met humanized mouse models. It's here again. Hemolysis. Hemolysis simply means our red cells getting broken up. When I was putting this lecture together, I was trying to look for some sort of animation to demonstrate hemolysis. And why, if hemolysis happens when it's not supposed to happen, it can be very bad. I've never used this before, so let's see how it goes. The best way I could think about it is palm nut. We all know palm nut, right? Yes? Yes? Palm nut is red. And we all like palm nut soup. But we also know that there's a kernel inside the core of palm nut. So hemolysis, when it's uncontrolled, is when somebody makes a benkwine by crushing the entire thing together. Not just the fruit. They crush everything together and make a soup with it for you. With the nuts, the kernel, all mixed together. That is not the way to prepare a benkwine. When red cells hemolyze, there is something in their core that is not supposed to be outside. It's called heme and hemoglobin. It comes out. And it's even more dangerous than the kernel in a palm nut. So I want you to think about how dangerous this intravascular hemolysis is. The other term I want you to be familiar with is acute chest syndrome and acute kidney injury. These are major complications of sickle cell. But it just occurred to me that we've been talking about sickle cell without me at least aligning you to where in our genome the problem lies. What you're looking at is called a karyotype. It's simply a lineup of all the chromosomes in our body. And in sickle cell disease, it's one chromosome, chromosome 11. That's where the problem is. That is because that is where this gene that I showed you before is located. It's called the beta globin locus. This is where we make hemoglobin. And in sickle cell disease, one change in the nucleotide, in the DNA, gives rise to the sickle hemoglobin. And that one change is responsible for erythroid cells, red cells, that are usually pliable. Red cells are very flexible because, we just talked about your blood veins. There are vessels deep inside your body that are very narrow. And so sometimes when a red cell is going there to deliver oxygen, because that's what red cell does, it has to bend and twist and go through that very small diameter of a vessel. But when the cells look like this, you can appreciate that it cannot bend. And so one of the major complications in sickle cell disease is sometimes the cells get stuck. It's like a traffic jam. They get stuck in a blood vessel, which means that all the organs downstream don't get any blood. Right? It's like going to airport and you get stuck at Shiashi. Complete jam. There's no traffic at 37. Same idea. So sickle cells get stuck to each other. They stick to the inside of the cell. We all know it's endothelium. And this is what causes what we call painful crisis. When they get stuck, they get damaged. And when they get damaged, they release this hemoglobin. These two things, cells getting stuck and breaking up, cause all these complications in sickle cell disease. So in part 2, what I was trying to do was let's develop an animal model for one of these conditions so we'll be better able to understand how we can treat it. The one that was of interest to me is a condition called acute chest syndrome. And it's exactly what it means. It's acute. It happens in the chest. And physicians, when they don't know what to say something, they just call it a syndrome. So this one was called acute chest syndrome for a long time. But the reason why it was of interest, it was the number one reason why sickle cell patients were being taken to intensive care. And there is no specific treatment. COVID came, everybody got something, at least to protect us from recurrent infections. Sickle cell disease, we've known about it. I share the names with you. Ancestral Ghanaians have known about it. Molecular geneticists have known about it. And the major complication in the lung, we don't have a specific therapy. Contrary to misconceptions, sickle cell mothers, there are several sickle cell mothers. There are women who have sickle cell disease who can carry pregnancy to term without any complication. But when they get complication, the number one cause of mortality is this acute chest syndrome that I'm talking about. This is a study that was done right here in Kolebu, where 87% of mortality of pregnant women was due to acute chest syndrome. When I became interested to try and figure this out, I had to think a little outside the box. You've already met heme. Heme is the byproduct of hemolysis. Around 2005, there were colleagues of mine who were using heme and injecting it into the belly of a mouse to induce hemoxygenase 1, which is a protective protein. I wanted to ask a very simple question. Knowing that sickle cell patients sometimes go to hospital with just a back pain or a leg pain, they have occlusion, and then before you know it, they have acute chest syndrome. So I had this idea that maybe what is happening is that traffic jam of red cells, the red cells all get crushed, and maybe heme is released in excess, more than the body can tolerate. And maybe that's what causing acute chest syndrome. Well, to answer that question, we could not inject heme into the belly, what we call intraperitoneal injection. We couldn't do that. I wanted to introduce heme directly into the blood circulation. So we did something that had not been done before. I had a postdoc who was very good. Mice have two large veins running along their tail. So we challenged sickle cell mice with acute elevation in heme, with a model that this is what is happening in sickle cell patients, except you can't really determine that in a patient. The outcome was quite remarkable. The petri dish on your left is the lung of a sickle cell mouse we have just challenged with saline, just saline. On the right is a mouse we have challenged with heme. I got so excited when we got this data, because we had taken a byproduct of the sickle cell pathophysiological process, challenged that with a sickle cell mouse, and triggered the mouse to have acute chest syndrome. We got so excited. This was eventually published in JCI, and this led for me to hypothesize a new model of acute chest syndrome pathogenesis, which is, we all know sickle cell patients get crisis. Those red cells, when they are caught in a trap, lies. When they lies, they release bad stuff like heme. Heme goes to the lung and causes a massive lung injury, in the process involving a signaling pathway called TLR4. I'm not going to bore you with TLR4, just that we're able to demonstrate that this actually happens in mice. And we're able to make sense of the sickle cell patients that die within 72 hours when they are diagnosed with acute chest syndrome. There are sickle cell patients who get acute chest syndrome within 24 hours, they are dead. Those patients we know have a massive hemolysis. So here we are taking a product of hemolysis, put it into a mouse, and these mice ended up dying so rapidly as well. If this is true, then it means that we can stop it, because the hemolysis is coming through here. We all hemolyze, so we have naturally occurring proteins that are supposed to protect us from hemolysis. One of them is haptoglobin, another one is hemopexin. All these proteins are already available and can be developed as therapeutics for acute care when sickle cell patients present with acute chest crisis. I can tell you there are companies currently developing plasma hemopexin, and we have worked with a company to develop recombinant hemoxygenase 1 as a means for acute management of acute chest syndrome. A lot of these haven't quite made it out yet, but the rationale makes perfect sense. Another model we developed recently was acute kidney injury. Here, we challenged sickle cell mites with a very modest amount of heme, and we're able to demonstrate that heme in excess is cleared by the kidney instead of the liver. Again, another model that is going to help us understand how we can improve the management of sickle cell disease. This data is simply showing you that whereas heme is going to the kidney in a sickle cell mouse, it goes to the liver as it's supposed to in a non-sickle cell mouse. And some of you may be familiar with this assay where we check how well your kidney is working. And you can see that in the AA mouse, this is again the mouse that is making normal hemoglobin, its kidney is perfectly fine. And the sickle cell mouse at baseline, even before any challenge, the kidney is not so good because you can see the 50% of clearance of this molecule. So this molecule is injected into the mouse and we see how quickly you can clear it. Whereas the normal mouse is doing it within 30 minutes, before 30 minutes, the sickle cell mouse is doing it much longer. And then once we challenge it, it's even much worse. So, Madam Chair, just to summarize part 2 of my work, we were able to develop the very first mouse model of the acute chest syndrome of sickle cell disease. Largely my work at Emory is where we did this. And then when we moved to Pittsburgh, we developed a mouse model of acute kidney injury. And I am pleased that we have developed yet a third model of cerebral infarct that is yet to be published. Now the final part of my lecture. The return home to help find a cure. You know, the return home officially supposed to have started 2017, but in 2013, 2014, my good friend here, Alice, is going to become very embarrassed. We started talking about how I can come back home. And we started doing work in KNUST, and that helped me to land softly because I had actually been doing work with colleagues in Konfanochi Teaching Hospital for at least 4 years before I eventually came. So Alice, thank you for that. The goal is to build capacity in translational genomics to allow for gene-based cures for sickle cell disease. My return home can be summarized in logos. This is also a time in my career when I discovered that I had a talent for coming up with logos. I can't draw, and so what I do is I hire an illustrator and I tell them where to put things. These 3 logos. The first one, Sickle Gen Africa. The second one, GA Genome. The third one, WAGMIC. All tell a story that builds on one project after another. We launched the Sickle Gen Africa project in 2018. But now you'll be familiar with this image. Building on my work in hemolysis, we wanted to ask this question. Does genetic variation influence the body's capacity to defend itself against hemolysis? And to do this, we needed to look in the DNA. And we took advantage of a program the NIH had developed called H3 Africa Consortium. And we applied for funding. This allowed us to basically set up this network with collaborators in Tanzania, South Africa, Nigeria, of course Ghana, and in Pittsburgh. This involved over 30 investigators with a simple goal of asking whether differences in our DNA contribute to our ability to defend ourselves against hemolysis. This is the experimental design of the study. This study is almost finished, and so you can focus your eyes to the very end. We are now looking at the data to see whether we can answer this question about whether differences in our DNA contribute to why one sickle cell patient is home playing video games without any problem, and another one is in hospital, and they are siblings. Because of the nature of this work, genes, I personally believe that the DNA that you have doesn't belong to you. The DNA you have was given to you by your mom and your dad. And before them, their mom and dad. So I consider DNA as part of our heritage. And the reason why I'm so excited to always work with traditional leaders is because I consider traditional leaders custodians of our heritage. So before we took a simple blood sample, we engaged the community. In Accra, in Lagos, in Kano, we held community engagement activities with sickle cell patients, parents, and explained to them what the Sickle Gen Africa project was all about. This helped us to recruit large numbers of patients. As of last year, the study is finished in terms of the enrollment. We have enrolled over 7,000 sickle cell patients in Ghana, Nigeria, and Tanzania. This is the largest cohort study of sickle cell disease in the world. The largest, by far. We are now in the process of sieving through the data. I've shown you two graphs. One is the serum concentration of hemoxygenase 1, and the other hemopexin. This data is of nearly 2,000 sickle cell patients. You see the community engagement, talking to the family, talking to the physicians, explaining what the disease is, the project is about and what it's not about, has really helped us a lot. We have finished the genotyping, and our colleagues in Pittsburgh are now asking the question, are there DNA sequences that explain why all of these are sickle cell patients? So if we look at the first one, is there a reason one patient has 1 nanogram and another one close to 100? We believe genetics will help us understand that. We leveraged the Sickle Gen project to start this Ghanaian Genome Project, which has 4 pillars. Community outreach program to sensitize Ghanaians on the role of genetics in health and disease. Work with traditional leaders to raise awareness of and screen for common genetic disorders in Ghana. Develop graduate programs, professional and academic, in genetics. And we are embarking on a whole genome sequencing of 1,000 Ghanaian children with sickle cell disease and other disorders. The GA Genome Project has been a major effort on our part to understand what is going on. Let's listen for a second.
So come do ye behind obia on to chanting la DNA test no obia sherbara cobia.
When we asked this question, taking a microphone to the streets, whether it was Tamale or Accra, the answers were basically the same. What we call the ordinary Ghanaian is not that ordinary. We had fishermen in Choco explain DNA. They may not use fancy words like deoxyribonucleic acid, but they knew what DNA was. We believe this effort of taking science to the street is going to be critical. When you start discussing with patients that you are going to do what? Change the sickle cell mutation in their body by infusing them with a virus, which is what CRISPR technology is. We are very, very passionate about this work. But engaging the community is not sufficient. You have to train the workforce that can explain this to people. And so last year we started the very first MSc genetic counseling program in sub-Saharan Africa. This is the first cohort. Next year, Madam Chair, will graduate them and hand them over to the Ghana Psychology Council for 1 year internship. Our goal is that within 5 years, we will have accredited genetic counselors in Ghana for the first time who will help in this genomic medicine that we are hoping to bring to this part of the world. In that effort, we are building infrastructure so we can do this work. We're hoping to complete a multi-purpose genomic building. When it's done, it will look like that. It's going to have a genetic counseling suite and all these facilities that will make it easy for us to begin to integrate genetics into the work that we do. I've already mentioned how important it is to us that we involve and engage traditional leaders, and we're doing that, as you can see from our front row. This was our visit to the Osumanche. At the height of Homowo, he still found time to talk to us. This was our visit to His Excellency, Sagnana. And all these folks, he gave us his compound for us to do the screen. We did the screen for sickle cell disease, prostate cancer, at the forecourt of the palace. And when I get a chance, I go on the airway and try to educate as best as I can. This was me here, Radio Tamale, early in the morning, here, Peace FM. The objective is the same. Promote awareness of genetic disorders so that we have to stop blaming old women sitting somewhere minding their business as the basis of a disease. So, Madam Chair, just to summarize part 3, we have established a unique biorepository for genomics and gene therapy studies in sickle cell disease, including assessing patient immunogenicity to potential gene therapy viral constructs. We have convinced corporate and sectoral stakeholders and traditional leaders of the importance of integrating genetics in healthcare services in Ghana. And as I showed you from the previous slide, we are building both the human as well as the infrastructure capacity through the center to overcome the developmental challenge of human genetic disorders in Ghana. And Madam Chair, if you permit me, I'll hold my last slide as really a last slide and just acknowledge those who have made this journey possible. You know, I got my interest in genetics from my A-level biology teacher, Professor Acquah. I don't know whether he's in the audience. He is? I don't see him. But this was my A-level biology teacher who was so much interested in genetics. I did my PhD under the mentorship of Mark Layton, Michelle Lalos, and Betty Pace. This is the list of folks who have worked in my lab. Countless collaborators. I apologize in advance if I've missed anyone. This is the list of collaborators in the Sickle Gen Africa Network. And the West African Genetic Medicine Center, headed by Melissa at administrative helm. We can't do any of this work in terms of the genetic counseling program, all this stuff we're doing. Somebody has to be pushing it from behind, and my team is fantastic at doing that. None of this is free. Most of my work has been funded by the NIH. Millions of dollars, NIH funding, so we thank the American taxpayer. More recently, the World Bank has funded our work, and that is because the government has sought funding from the World Bank to support our work. So, Madam Chair, as we close, I want us to come back to the first slide. The inaugural lecture is a journey with sickle cell disease around the world and a return home to help find a cure. You may recall that I identified a challenge that we have. The success of newborn screening and hydroxyurea therapy means that we're going to have more sickle cell, people with sickle cell living. So the challenge here is to find better ways to manage and ultimately cure this disease. I believe The scientific journey I have shared with you helps a little bit for us to overcome this developmental challenge. And the answer is in the 3 logos. The Sickle Gen Africa project has allowed the University of Ghana to have the largest biorepository of sickle cell disease in the world. It's sitting right here in the University of Ghana. The GA Genome Project is making genetics less scary. Maybe it was actually less scary, we just didn't know. People know about genetics. They know about inheritance. That lady we met in the market in Tamale, I only showed you a small piece. She gave us a long history about inheritance. The GA Genome Project will continue to awaken the consciousness of the Ghanaian about the importance of genetics in health, well-being, and disease. That's what we want to do. And then, of course, WAGMIC, the center that now is growing concrete roots in the University of Ghana, and we're developing programs, which means that we are here forever. Long before I'm gone, somebody else will take over and maintain the sustainability of the programs that we have built. So, Madam Chair, these 3 logos or symbols give me hope that the state of our preparedness to improve management of sickle cell disease in Ghana is good. Thank you.
Madam Chair, ladies and gentlemen, I believe you all side with me that Prof has broadened our perspectives and horizon. In fact, I have learned today that DNA is part of our heritage. And I'm particularly thrilled by the fact that moderate exercise is good for sickle cell patients. I remember back in school, we used to hold sickle cell patients like eggs. We were afraid to go near them or to tamper with their lives. So I'm very happy to learn that they can do moderate exercise. And I believe you're also happy to learn this and many more from Prof. Let's give it to him again. Distinguished ladies and gentlemen, next, the Ghana Dance Ensemble treats us to the Asafo dance. Asafo was a warrior dance used to lead armies into battle among the Akans. The dance was also performed in jubilation after a war or battle is won. According to the Ghana Dance Ensemble, the work of an academician is a battle. Therefore, when a momentous battle, such as the attainment of the rank of a full professor, is won and crowned with an inaugural lecture like this, it is very right to celebrate with an Asafo dance. Asafo dance is also very apt for this occasion as it reflects the ethnic background of Prof. Ofori Acquah. Ladies and gentlemen, the Asafo dance from the Central Region.
School of Performing Arts, together with its team of performers.
Let's give them another round of applause. Madam Chair, ladies and gentlemen, it's now time for presentations. But before we move on to that, please permit me to acknowledge some personalities that have joined us this evening. We are lucky to be joined by our former Vice Chancellor, Professor C.N.B. Tego. And we are also happy to have Professor Ellis Owusu-Dabo, Pro-Vice Chancellor from KNUST, together with his wife. We also have here in our midst Dr. Darius Osei, CEO of University of Ghana Medical Center. And I think they've been mentioned over and over, and so at this time when I mention them, I will implore them to give us a wave. We have representatives of the Osu Traditional Council, and then we also have the Sangna Na and his entourage here with us. I'm also happy to inform you that we have Dr. Kwabena Bempatando, Deputy Director General of Ghana Education Service, here with us. And we were also joined online by Professor Lydia Aziato, Vice Chancellor of the University of Health and Allied Sciences. I'll now move on to the presentation, and I believe that we're going to see a lot more people who joined us this evening. Professor Ofori Acquah is coming from the College of Health Sciences. As we've learned this evening, he's the director of the West African Genetic Medicine Center and a former dean, the immediate past dean of the School of Biomedical and Allied Health Sciences, both of which are established in the College of Health Sciences. And so I'll first of all invite the College of Health Sciences to do their presentation, and then we proceed from there. The School of Biomedical and Allied Health Sciences should please be on their way. School of Biomedical and Allied Health Sciences. And the Department of Medical Laboratory Sciences should get prepared. Department of Medical Laboratory Sciences. Department of Medical Laboratory Sciences.
In the West African Genetic Medicine Center, Prof Center. While the West Africa Center for Cell Biology and Infectious Pathogens gets ready. WAGBIP, please get ready. While we are waiting for WAGBIP, let me share this joke. In my office, we call these centers the WA people. So we had the West African Genetic Medicine Center come, and we are waiting for the West Africa Center for Cell Biology of Infectious Pathogens. The other WA people are the West Africa Center for Crop Improvement, that's WACI. We invite research networks and collaborators, Sickle Gene Africa and Ghana Genome, GA Genome, to get ready. Research networks and collaborators, GA Genome and Sickle Gene Africa. While they are here, University of Ghana Medical Center should get ready. University of Ghana Medical Center. University of Ghana Medical Center, UGMC. Sickle Cell Foundation of Ghana, please get ready. Sickle Cell Foundation of Ghana. And Adisco Old Boys, please get ready. Adisco Old Boys. Adisco Old Boys. I was hinted by Prof that when you get here, you sing your anthem.
Those who are building the school without faith, a blessing upon us.
but not least on my list is the family of Professor Ofori Acquah. I now invite forward the family of Prof. Ofori Acquah to make their presentation. We thank you all for your presentations. Yes, please. Prof, you can take your seats now. Ladies and gentlemen, it's now my duty to invite our chairperson, Professor Nana Apia Amfo, to deliver her closing remarks. Please let's welcome her to the podium.
Thank you, Professor Ofori Acquah, for giving us a very insightful lecture, taking us through lessons on sickle cell disease, genetics, genomics, name them. And thank you for bringing the Santa Clausians here. We must admit that they have the best anthem and the nicest voices. I needed to say that to win some points. Right, so indeed, there's been a lot that we have learned this evening from the lecture. Professor Ofori Acquah told us the state of sickle cell disease in Ghana. He ensured that he acknowledged the pace setters, the forebearers, the various partners and institutions and others who have worked in this area. And I was particularly happy and encouraged by the advocacy that has been done that has ensured that newborn screening has increased, that has ensured that hydroxyurea is on the essential drug list, and it's available for people living with sickle cell diseases and helping with the management of the disease. And then he took us to the lab. I had warned him before the lecture to keep it simple for ordinary people like me to understand, and I think that he tried to do that. So he did expose us to the various terms, the fetal hemoglobin, the beta globin glucose, the endothelial cells, the hemolysis, and all of those. But most importantly, he gave us an idea of what he has done in the laboratory from London, England, through to the US, and having collaborators from all over, and what it has resulted in. So at the end of the day, one would ask that what has been the benefit of all of these? As we say in Ghana these days, ne kra kra ne sing. Well, there's been a number of things which have resulted from all of these efforts. The first I'd like to talk about is the public health education and the awareness campaign. And so now we know a lot more about this disease. There's been the advocacy that I talked about. There's a lot more awareness all over, and it is continuing. The other thing too that has come up is the training of a new cadre of health professionals in Ghana, not just in Ghana, but in the sub-region in genetics. I am particularly happy about the multidisciplinary work that is ongoing. And so the training is not restricted to traditional health professionals, health workers, but there is a recognition that the health sciences need to coexist with the other sciences, such as the social sciences. And so we're seeing the training of the first group of genetic counselors, and my understanding is that these are mostly psychologists, and they are being trained to help us all manage this particular disease, which is of concern tonight. So there's a general capacity building in genetic health across board. I'm also excited about the recognition and the collaboration, the recognition of traditional or indigenous knowledge systems. When he started his lecture, one of the first things he did was to tell us about the different names of the disease in the different languages that we have in Ghana. And once we have these names, we would say in linguistics that once we have these lexicalized in the various languages, then there's the recognition that the local community, they know about the disease and they have their own understanding of these diseases. And we need to recognize these because it helps a lot with the public education. We need to know that we are not starting from scratch. There are things that already exist in the community, and we need to take that and then build on that from there. It's also worthy of mention the collaboration with industry partners, the pharmaceutical companies, and so on, all in the bid to find a cure, a lasting cure to this disease, not just to manage it, but actually to find a lasting cure to this. And so as I sat down and I listened to him, I thought that what's the end process of this all? I coined an acronym, TSS, and that is taking science to the streets. And that is what our professor has demonstrated. He's been to the lab, but he's not just stayed in the lab. He's gotten out to the streets. He's gotten to the radio stations, he's gotten to the communities, working together with traditional leaders, working together with the youth, working together with people within the community to ensure that we have a better understanding of this disease, we manage this disease, and ultimately find a cure to this disease. Professor Ofori, Acquah, I'm very proud of you. This is the kind of academic that we are expecting to be producing in our institutions. He has demonstrated that science is not just for itself. Science has to indeed benefit our communities. And we thank you for showing the way. I know that many more, others have done this. You have demonstrated to us tonight, and many more will follow after you. So thank you all for coming this evening. We have some more inaugural lectures outlined. I believe that the Director of Public Affairs will tell us more about that. But when you hear the announcements, do come listen to us, come and support us. At the University of Ghana, we do good work, we do science, and we take science to the street. Thank you and have a good evening.
Well said, Madam Vice Chancellor and Chair. We thank you also for your words of wisdom. Yes, indeed, we'll continue to take the gown to town by sharing the cutting-edge research done by our faculty. We thank you so much. I'm adding my voice to the Vice Chancellors to thank you so much for joining us, both physically and online. As she rightly said, we have a lineup of lectures, and I'm excited to announce the next inaugural lecture, which will be delivered by Professor Gordon Awandare. You can see the flyer on the screens. Professor Gordon Awandare is our Pro-Vice Chancellor for Academic and Student Affairs, and also the Director of the West Africa Center for Cell Biology of Infectious Pathogens, one of the WA centers. Okay, so his topic is how our immune system acquires tolerance to malaria and helped us to survive COVID-19. I think you wouldn't want to miss this for anything. So we expect you on June 18, 2023, here at this very hall for Prof. Awandare's June 8th at this very hall for Prof. Awandare's inaugural lecture. You are all most welcome. I am happy to announce that we are in our 75th anniversary, and the celebration is ongoing. You can see beautifully displayed there our 75th anniversary logo. Please stand by it, take photographs, and share on your platforms, and spread the word about our 75th anniversary celebrations. We have a lineup of programs that we are doing in line with our 75th anniversary. We publicize them on our website, social media platforms. Please look out and join us in celebrating our achievements. I would like to take a couple of announcements on refreshments. We have refreshments for senior members and our guests on the wing to my left, the north wing. So all senior members and our guests should join us, should join the platform party on the wing to my left for refreshments. Staff and students will take their refreshments at the AA Kwapon Quadrangle, which is the forecourt of the Great Hall. At this point, distinguished ladies and gentlemen, please let's rise for the university anthem. You will find the lyrics on page 16 of the brochure and also on the screens. Thanks very much. Please remain standing as the platform party recesses.